All right, so we'll do a quick review here, and we're about done. By the end of this period, we really only have one slide of notes left. Glycolysis, you've got Krebs cycle. We started yesterday. Did Krebs and electron transport. Krebs cycle, the one you need to know the least detail about, right? It's basically that we're breaking pyruvate down step by step into carbon dioxide, which you breathe out. In the process of that breakdown, we get a lot of babysitters. We get a lot of high energy electron carriers called NADH and FADH2. You also get a couple of ATP. The pyruvate and the FAD and NAD plus and the ADP plus P are the reactants. Carbon dioxide and the carriers and the ATP are the products. Then your big moneymaker being the electron transport chain. That's where all your NADH from glycolysis and Krebs and FADH2 from Krebs gets sent. In that process, as I crudely drew yesterday, we can see that the electrons jump along that chain of proteins. The work that gets done because of that energy is protons being pushed into the intermembrane space. That's this space between the two mitochondrial membranes. And that those protons leaking back through, because they really want to, spins the ATP synthase and powers the production of a buttload of ATP. Then you learned that if something wasn't here to pull these electrons off the end of the chain, the electrons would back up and shut the chain down, which would be bad. You would lose that 32 ATP. So to keep electron transport going, you bring in oxygen. Your circulatory and respiratory systems work together to deliver oxygen to every cell in your body, and that's forming water, what keeps your electron transport chain going. It's your final electron acceptor for the electron transport chain. Got all that? Okay, so today we talk about what happens when there is no oxygen, which means there is no electron transport, which means Krebs shuts down and you're left with glycolysis to keep going. But first, let's draw a picture. This is one of those you're going to want to wait until I finish. It's a little busier than the photosynthesis one, a little less tidy. Here we go. Mitochondria here. Membrane on the inside. Glycolysis happens in the cytoplasm, Krebs cycle in what we call the matrix, and electron transport along this inner membrane. Glucose comes in glycolysis. 2 ATP gets spent. Water is given off. 4 ATP are produced along with NADH. You can ignore that FADH2 for now. That actually comes from the Krebs cycle. I just animated it wrong. But what the glucose turns into physically in glycolysis is 2 pyruvic acids, which get sent to the Krebs cycle. Krebs cycle spins, creates 2 ATP, gives off CO2, which you breathe out, and sends a buttload of NADH and FADH2 to electron transport. Good so far. Electron transport takes that NADH and FADH2, teases those electrons down that chain, making ATP. Needs oxygen to pick the electrons up. It creates 32 ATP for glucose and water as a byproduct. Now, optional for you, but it is important to note that the NADH and the FADH2, once they're stripped of their electrons, are going back to glycolysis and Krebs to pick up more electrons. Those are actually reactants in glycolysis in the Krebs cycle. Makes it extra busy, I know, but it's fairly important to go. This whole thing is, let's see, there's a couple ways to put it. Some would call it oxidative cell respiration, aerobic cell respiration, or if you're kind of animal and plant centric like we tend to be, just cell respiration. But This does not include what happens when there's no oxygen, which is sort of another kind of cell respiration, I suppose. Yes. Let me get rid of those two yellow arrows for you. Help? Mm-hmm.
What does the electron transport chain send back to Krebs cycle?
NAD plus and FAD, the D electron carriers, right? The electron transport chain strips the electrons off of them, and they get sent back. So yeah, there should be an FAD here as well.
Is it between like the matrix and
Yeah, so the thing I drew here, notice it was kind of linear. Basically, that's saying that the electron transport chain is actually embedded in this inner membrane. The proteins that the electrons jump across. And what's happening is that the protons are being pushed into this space here and then being allowed to leak back through here to make ATP. That makes sense? Any other questions before we talk about not having oxygen? Because remember, without oxygen, that'll mean this backs up and shuts down. And it'll mean you can't get rid of NADH and FADH2. Nothing will be stripping the electrons from them. And so you won't be able to send those back here and here. So they'll shut down as well, because NAD plus and FAD are reactants in those two processes.
How fast does this process happen?
Which process?
Like that whole thing.
Electron transport is slower than glycolysis. I can tell you that. As far as real time, I don't have a number for you. Okay, so the answer to what you do when oxygen isn't there is fermentation. Because as soon as electron transport shuts down, glycolysis and Krebs are only going to work until the available babysitters, that is NAD plus specifically, and this alternate pathway is basically going to do the job of the electron transport chain in terms of stripping the electron off of NADH. Krebs is gone, but glycolysis is fast enough to keep you going for a while if you can just keep supplying NAD+. And so fermentation is what allows NAD+ to be recycled. It doesn't make any ATP on its own. Fermentation is not an energy generating process. It just allows glycolysis, which is to keep happening. It turns out there are two kinds of fermentation. Alcoholic fermentation, which is done by one particular kind of fungus called yeast, one of the few one-celled fungi, and many bacteria. And the equation for alcoholic fermentation, along with structural formulas, because I think they kind of come in handy now to see what you're dealing with. The products of glycolysis, right? Pyruvic acid and NADH, the major products of glycolysis besides ATP, which, you know, those go in. And what comes out are ethyl alcohol, carbon dioxide, and NAD+. Here are the structural formulas of pyruvic acid and ethyl alcohol.
We need to know that.
Not really, no. But I think it's helpful to see that what you're doing with pyruvic acid in alcoholic fermentation is basically just chopping off one of the carbons and turning it into CO2. Most of you already know a little bit about this because of the commercial uses of yeast. What do we use yeast for in our society? One use is bread, absolutely, because bread is a mixture of flour, which is ground wheat, and water that turns into a paste, and then you bake it. Well, if you did that without any yeast, what you get is something that would break your teeth. Right? Unless you flatten it out real thin, in which case you get crackers. What you need is air bubbles in that paste to kind of puff up and make it soft and pillowy on the inside and nice and crusty on the outside. And yeast is the way you make that happen. In fact, it kind of does it for you. It's a living thing, right? You mix yeast up with the flour paste, and the yeast wakes up. You get it dried. It's like suspended animation, and it wakes up when it gets wet and starts eating the sugars from the flour. It starts stripping glucose off amylose and creating alcohol, which is going to bake off in the bread anyway. But more importantly, little bubbles of CO2, little bubbles of carbon dioxide in that paste. And flour is such that it has a protein network that'll hold those bubbles. And then when you bake the bread, the bubbles get even bigger because the CO2 expands. And that's what makes bread like edible and soft on the inside. Absolutely. What's the other thing we use yeast for? Well, nothing that you mess with yet, but yeah, beer, wine, spirits. Yeah, the stuff that makes adults stupid, right? The yeast that you put in sugar water, depending on what kind of sugar, tells you what kind of alcohol, right? If it's grain sugar, if it's malt sugar, it's going to be beer. If it's grape sugar, that's wine, right? But you just mix the yeast up with the sugar water, and it does its thing eating the sugar and makes alcohol and CO2. It happens to be ethanol, which is that stuff. And then, you know, if you cap it, if you contain it before all the CO2 is bubbled off, that can carbonate, right? That's why beer is fizzy, or can be, or champagne, right? But if you let it all bubble off, then it's not, okay? So that's obviously not what you do when you run out of oxygen. When you exercise, your muscles run out of oxygen, and they have to switch to a different pathway. But it's not that, or, you know, you get drunk every time you go out running. It'd be a whole different crowd at the track. No, you do something different, which is called lactic acid fermentation, which I bet, yeah, Ben's shaking his head like, yeah, my coach talks about that crap all the time. Yeah, lactic acid is what because you can't just split off of CO2 and make ethanol and CO2, because not only would you be creating toxic alcohol in your bloodstream, but you'd be creating bubbles, which would probably also kill you. So instead, you convert your pyruvic acid with a little more work. You convert it into something called lactic acid, which is only slightly toxic, unlike alcohol. Changes pyruvic acid into lactic acid, which works great. And it's fast, because glycolysis happens super fast. And you can do a lot with lactic acid fermentation. You can sprint 100 yards, you can power lift hundreds of pounds, jump, I don't know, what's a good long jump track? A good distance. I have no idea. 15 feet? 20 feet? Anyway. all those kinds of hard as you can, maximum effort for a short period of time. That's all anaerobic stuff. That's all going to be lactic acid fermentation. But not all roses. You can do it, but you can't do it for long because lactic acid builds up. Lactic acid hurts a little bit in your muscles. Creates that burn that you feel when you're going hard. Eventually, creates an acidic environment in your muscles where your muscles don't even work anymore. You will give out if you just go full tilt for as long as you can. More likely, though, you'll just stop at the end of your sprint and do what? What happens after a sprint? What do you see people doing?
Well, I know, because you throw up after.
Yeah, maybe throw up, but definitely the classic kind of hand on knees, sucking wind, right? Like you got, well, this is why. It takes oxygen. This is the other thing you use oxygen for. The main thing is electron transport. The other thing is if you switch to lactic acid fermentation, it's oxygen that comes and grabs lactic acid and takes it away from the muscles, gets it out of there. And it doesn't happen super fast. So what you build up is called oxygen debt, which is why the post-sprint position is this, right? It's why you can't do it forever. Gotta wait for your bloodstream to catch up and deliver enough oxygen to get the lactic acid out there. It's also why Ben's coach tells him to lay on his back and bicycle his legs in the air to drain the lactic acid out of his legs. Am I right?
I haven't done it.
Adams?
Yeah.
Which I think probably works. Gravity. Yeah, because lactic acid, if you don't get it all out of there, especially if you've gone a little further, a little harder than you normally do, your body doesn't get all the lactic acid out of there, and the next day, man, that hurts to have residual lactic acid losses. Equation-wise, we are looking at, again, pyruvic acid and NADH going in. But this time, instead of CO2 and alcohol, we just have lactic acid itself. Formula-wise, just to get a sense, that's what these look like. Really, we've just changed one double bond. But it was enough to feed an electron or to strip the electron from NADH and create an available babysitter to go back to glycolysis. Structural formula? I won't make you draw the structural formula. Other questions? Probably good to get a look at this on the overall diagram. So I made one that kind of shows what happens. Everybody got this? More time? Okay. So here's normal, right? This is what you normally do. Your muscles usually do glycolysis, send the NADH to electron transport, Krebs cycle, send the NADH and FADH to electron transport. Well, without oxygen, ETC shuts down. Which means Krebs cycle shuts down, which means glycolysis would shut down because you're not getting NAD plus sent back. And so now you're not getting these ATPs. We really need these ATPs. So instead of sending NADH to the electron transport chain, glycolysis sends it to fermentation, sends pyruvic acid to fermentation, which recycles the NAD plus and then produces either lactic acid or CO2. So basically, we've eliminated the mitochondria for now. We're just going on glycolysis. Can't do it for long because of lactic acid. And a bacteria or yeast can't do it too much either. The reason they can do it with something toxic like alcohol is that they're one-celled organisms, and they're floating in basically soup, right? They're floating in fluid. And for a while, they're fine pushing that alcohol off into the soup they're floating in. However, if too much alcohol builds up, they will die, which is why you don't find certain alcoholic beverages above a certain percent alcohol, because the yeast die at that point. Right? You don't get much above 10 to 13%. Beer is usually 10, but that's hot, right? You got to work to get them to survive that long. So they're one cells, they get away with that, whereas not so much with you. Yeah.
How do you know which one's good?
Whether it's lactic acid or alcohol, what kind of creature you are. If you're you're only going to do lactic acid. Yeah, you can't do alcoholic fermentation. If you're a bacteria or yeast, you're only going to do alcohol. Not like you choose one or the other. I mean, you don't choose. Like you do one or you do the other. You're programmed out. Good? Questions?
So like after this whole like fermentation process happens in the oxidant,
Uh-huh.
so like does that NAD plus then would that go like the electron
No, NAD plus goes back to glycolysis. Gets turned into NADH again and then goes back to fermentation. Until the lactic acid builds up so much that you have to stop, that's what's happening. Now, we'll talk about exercise tomorrow, but like if you're pacing yourself right, if you're a distance runner, if you're in it for the long haul, what happens is, you do some fermentation at first. There's no way around it. But you go slow enough and you pace yourself enough that eventually this catches up and starts working. Yeah, because you increase your blood supply and your heartbeat and all that. All right, we are looking at a test on Tuesday. Tomorrow's the last little bit of notes. Here's sample essays for you. They are due on Monday. Remember that tomorrow is like a Friday. If it helps, after this, we're going back to chapters 3, 4, 5, and 6, which are ecology. Ecology is not easy, but it's different. It's not all about chemicals and formulas and counting things and drawing pictures, right? It's much more concept-based. It'll be more kind of writing type essay questions. It'll be a relief for lots of